So from this image, can anybody guess what is the class on today? Cell injury. Okay. So the competencies we'll cover today are 2.1 and 2.2. So majorly the key points are going to be cell injury, a little bit about it. Then etiology of cell injury, pathogenesis, reperfusion injury and morphology of cell injury, majorly morphology of irreversible cell injury and a small recall session. So what is cell injury? So cell injury is defined as the effect of a variety of stresses due to etiological agents which a cell encounters resulting in changes in its internal and external environment. So considering this as a cell in presence of stressor agents like bacteria or any physical trauma or anything for that matter, the cell will undergo certain changes both within itself and also in its surrounding stroma. So those changes together, they are known as cell injury. So what are the cellular responses to cell injury? So under stress, the cell initially will try to cope with the altered functional demand. How does it cope? It undergoes some adaptations like atrophy, hypertrophy, hyperplasia, dysplasia. So these adaptations in details will be dealt with in the subsequent classes. Now, when the stress persists and it is present in its mild to moderate form, then the cell undergoes some reversible cell injury. So what does it encompass? It has degenerations, some subcellular alterations and intracellular accumulations. Now when the stress is removed at this point, the cell will limit itself to the irreversible cell injury. But if the cell injury persists from this point onwards, then, or if it is severe stress, then the cell will undergo irreversible cell injury resulting in cell death. This is clear? Yeah? So to put it in a diagrammatic format, which you have to remember for your exams. So normally the cell is under homeostasis, right? Normal functioning. Now when stress comes in, it will undergo some adaptations. Like we read, right? Hypertrophy, atrophy, dysplasia, metaplasia, all these adaptations. So when the cell has an inability to adapt, it will undergo cell injury. Okay? So this cell injury, in presence of mild and transient stress, it is going to be reversible. And when the cell undergoes reversible cell injury, it can always go back to its normal functioning. But if the stress is severe and progressive, then it will be irreversible cell injury, which will have two forms. It will either result in necrosis or apoptosis, both resulting in cellular death. This is a very important diagram. You will have it in your textbooks as well. Please remember it. Okay? So now what are the general mechanisms of cell injury? That is, what all organelles or what all parts of the cell are usually affected by cell injury. So four very interrelated cell systems are particularly vulnerable. The first thing which gets affected is the membrane. So membranes, it can be either of the cell, entirely cell itself, or it can be of the organelles as well, like that of mitochondria, lysosome, Golgi apparatus, all of that. Then secondly, aerobic respiration is affected. So what happens if aerobic respiration is affected? What happens? What gets accumulated in the cell? Yes, lactic acid will get accumulated and there will be disbalance of the pH. Then protein synthesis will be also affected. So proteins, what are the proteins? They can be enzymes or even structural proteins. Okay? And the last part is the genetic apparatus consisting of the DNA or the RNA can also be affected. So what are the etiologies of cell injury? So they can be broadly divided into genetic causes and acquired causes. So the best example of genetic causes are syndromes or inborn errors of metabolism. Do you guys know what are the inborn errors of metabolism? Or any idea about it? Yeah? In biochemistry, you must have dealt with it, right? Anybody would like to volunteer with the answer? Yes, correct. Yeah. So what happens in inborn errors of metabolism is that there is accumulation of misfolded proteins within the cell. So what do these misfolded proteins act as? They act as toxic or injurious agent which hampers the function of the cell, resulting in cell injury. Now what are the acquired causes? We have a long list. It has hypoxia, ischemia, some physical agents, chemical agents, microbial agents, some immunologic agents, nutritional derangements, aging, psychogenic diseases, iatrogenic factors, and idiopathic diseases as well. So this is also a question, etiologic factors of cell injury. So the most important amongst all of this and the most common is hypoxia and ischemia. Okay. So hypoxia is the most common cause of cell injury. Can anybody tell me the difference between hypoxia and ischemia? Yes, please. Yes, you are very correct. So, as your colleague said, hypoxia means there is decrease in the oxygen concentration at the target organ, and ischemia means it is a case of decreased blood flow to the target organ. Okay, so when you think of hypoxia and ischemia, which one do you think is going to cause a more severe form of cell injury? How many of you think it's hypoxia? Put your hands up. Nobody thinks it's hypoxia. Okay, one person. Come on, volunteer. Okay, 2, 3, we've got 2 more. 5. Rest of you, what do you think? It is ischemia. Actually, the answer is ischemia. Why so? Because hypoxia, although it is a state of reduced oxygen supply, but some of the oxygen is still going to the target organ. But ischemia means there is acute cutoff of the blood supply. So we'll have almost no oxygen going to the tissue. So there are 2 reasons. One, it can be because of reduced blood supply to the cells due to interruption in blood flow leading to ischemia. Or it can also be due to defects in oxygen carrying RBCs, or due to some heart diseases, lung diseases, or due to increased demand from the target tissue. Now an example of the interrupted blood flow. So this is a normal artery, right, which has a patent lumen, and the picture beside shows us an artery with an atherosclerotic plaque. So what happens in presence of atherosclerotic plaque? The lumen narrows, right? So if the lumen is narrowed, then the blood flow to the organ is impaired. So which organ do you think is most affected? Heart. Correct. Now we have intrinsic factors of organs affecting the blood flow. So like in case of heart, we have ventricular hypertrophy or cases of dilated cardiomyopathy. So in both the cases, what happens? The contractility of the heart is affected. So if the contractility of the heart is affected, then the pumping capacity is affected. So if the heart doesn't adequately pump the blood, then there is hypoxia of the target organs of the peripheral tissue. Yes, yourself, Miss. Last bench, you said. Please come and sit in the first bench. Come up. Next, abnormal RBCs. So what is the normal shape of RBCs? Biconcave. So what does it facilitate? The normal shape of RBCs, it facilitates the maximum oxygen carrying capacity of the RBCs, and also it makes it safe from hemolysis within the circulation. So in cases of abnormal shapes of RBCs, we have 2 issues. One is that the RBCs are unable to carry the oxygen efficiently, and secondly, they become very prone to lysis within the circulation. Okay. So where do RBCs get destroyed? Spleen, correct. So there is an increased sequestration by the spleen in presence of abnormal shapes of RBCs. Now next, lung diseases. So when the parenchyma of the lung is affected, what will happen? What happens in lung? Oxygenation happens, right? So if the lung has a parenchymal disease, then what will happen? The oxygenation is going to be affected, again resulting in hypoxia. Now physical agents, it's simple. We have cold trauma like frostbite, then burns, radiation trauma, and even electric burns, which can lead to cell injury. Chemicals and drugs, this is also important. Please remember the names. Okay? Poisons like cyanide, arsenic, then strong acids, alkalis, some pollutants, insecticides and pesticides, even oxygen at high concentrations can cause cell injury. Then hypertonic glucose and salt, social agents like alcohol and narcotic drugs, and even some therapeutic drugs can lead to cell injury. So are you aware of any drug which affects the liver at very high concentration though, but it's a very commonly used drug? Sorry? Paracetamol, yeah. Acetaminophen, it can cause lethal liver toxicity, but it's at very, very high doses, so it's almost a very safe drug to use. So microbial agents like bacteria, viruses, fungi, protozoa and parasites. So any organism which has a infective capacity can cause cell injury. Immunologic agents. So immunity plays a double-edged sword. What does this statement mean? One is that the immunity, our body's immunity helps us to ward off all infections, right? So that is how we are healthy. Now how does it affect the other way around? One example is given right away here, autoimmune diseases. So when our own immunity goes against our own body, but doesn't recognize self, then it becomes autoimmune. Other causes. So what happens when our immunity is functional and is fighting off the infection? We have certain agents which are being involved, mostly cytokines. Okay. So these cytokines, they not only affect the organisms or the infective agent, but they also affect the normal cells. So do you know any example? Like I think some few years ago we had a massive outbreak of COVID-19. What followed? Majority of the deaths happened due to what? Do you remember the term? Something called cytokine storm? No? Okay, so cytokine storm is one of the major causes of death during the COVID-19 pandemic. So what happened was, the cytokines that our body was producing to fight against the COVID-19 had its deleterious effects on the normal functioning of the body as well. Okay, so this is one example you can remember for our immune response fighting against our own body. Now the pathogenesis of cell injury. So in pathogenesis, we have to know that if a normal cell is affected by any of the etiologic agents that we just studied, it can either cause reversible cell injury or irreversible cell injury. So what happens if there is reversible cell injury? How does the cell react? How does the cell react? It goes back to being normal, that is, it undergoes repair and healing. But if there is irreversible cell injury, there is cellular death. So the outcome of the cell injury, it depends on certain characteristics of the injurious agent and that of the target cell as well. So what are the factors of injurious agents? The type of the injurious agent, duration of exposure and the severity of exposure. And the target cell characteristics includes the type, the status and also the adaptability of the cell, which affects the outcome of the cell injury. So how does the type, duration and severity affect? So small doses of the chemical or the physical agent usually results in reversible cell injury and large dose results in irreversible cell injury. So very common example for this you can take like when you are exposed to hot boiling water. So if it's a droplet or 2 or 3 droplets, how does our body react? We get blisters, right? Maximum. We get blisters which gets healed within a week or so. Now just imagine if a bucket or you are exposed to a big volume of the boiling water, what happens? We get a scalding injury, right? Scalding burn. And it doesn't take a week or so, it takes a longer duration to heal, sometimes also resulting in contractures. So, if it's a short or a small dose, then it is reversible. If it is a severe dose, then it is a irreversible cell injury. Next is short duration of ischemia will result in reversible cell injury, and long duration is irreversible cell injury. So if we take the case of, transient ischemic attack, any idea what transient ischemic attack? What that is? Which organ does it affect? No idea? Yeah, it affects the brain, right? So transient ischemic attack, as you can know from the name, it happens because of a very temporary and a very short span of interruption to the blood flow to the brain. So it usually has certain symptoms, but they usually resolve. So we do not have a long-standing cell injury. But if we have ischemia for a really long time in the brain, what happens? What do we get? Cerebral, ischemia is the cause, the effect is infarction, okay? And if there is cerebral infarct, we get long-standing complications and symptoms which doesn't resolve easily. Now, how does the type, status and adaptability of the target cell affect? So, if the cell is highly susceptible to hypoxia, then there is early cell injury. So, an example of this is the difference between skeletal muscle and cardiac muscle. So, skeletal muscle can withstand hypoxic injury for a long time. So, we all know that athletes go for running and lot of hiking, or whenever we go for any activity at challenged situations, there is hypoxia to the skeletal muscles, but it's not like the muscles die right away, right? So, there is deposition of lactic acid, not anything beyond that. But if the same thing happens with our cardiac muscle, and if there is hypoxia persisting beyond 20 minutes, then the cardiac muscles undergo irreversible cell injury. Okay? So now we'll know the principal mechanisms of cell injury and their biochemical and functional effects. So before we start this, can anybody recall what all the organelles or what are the parts of the cell that get affected during cell injury? Cell membrane, then nucleus, ribosomes, endoplasmic reticulum, that will come in the membrane. Okay, membranes of either the cell or the organelles. That becomes the first. All of these organelles did not affect directly, the membranes are affected. So the membrane will be affected, it can be either of the cell or the organelles. Then second, what is the ATP generation mechanism in a cell? Something with some kind of respiration, aerobic respiration. The third is, how is this protein affecting the cell? What does it include? Within a cell, enzymes and structures. So the protein synthesis will be affected, and fourth is the DNA or the RNA. Okay? Please remember this. So now, when we take ischemia as the stressor agent, the first organelle which gets affected is the mitochondria. This is very important, okay? Please listen very carefully. So the mitochondria is affected and there is reduced oxidative phosphorylation resulting in decreased ATP formation. So what happens if there is decreased ATP? One is that the sodium pump functionality will be reduced. Second, there will be increased anaerobic glycolysis. Okay? And the third is there will be detachment of the ribosomes from where? Yeah, from the rough endoplasmic reticulum. So what happens if there is decreased functionality of the sodium pump? There will be increased influx of the calcium, water and sodium ions. Remember it. So there will be entry of, what are the three things? It's just written here. Calcium, water and sodium within the cell. Okay? And there will be increased efflux of the potassium. So potassium is going to go out, sodium, water and calcium is going to come inside the cell. So this will result in swelling of the endoplasmic reticulum, cellular swelling, loss of microvilli and formation of blebs. Now what happens with increased anaerobic glycolysis? There will be increased lactic acid, reduced pH resulting in clumping of the nuclear chromatin. And with detachment of ribosomes, there will be reduced protein synthesis, and if there is reduced protein synthesis, there will be reduced forms of lipoproteins which are required. So if with reduced lipoprotein formation, there will be accumulation of lipid or lipid deposition. Okay, this is clear? Is this clear? Yeah. So up till this part, the changes that happens up till this part are all reversible. Okay. So if we remove the stress from this point onwards, like if the ischemia is corrected by now, then the cells can go back to their original functionality. At this point, it is still reversible. But if the stress region or the ischemia still persists and we are unable to correct it, then the changes of irreversible cell injury are going to take place. So, we've read like from the previous slide that there is going to be increased influx of the calcium. So, the extracellular calcium is going to come inside the cytosol. And the membrane of the mitochondria and the endoplasmic reticulum is also going to be affected. So what happens? The calcium ions from the mitochondria and the endoplasmic reticulum also move out into the cytoplasm. So all in all, what happens is there is an increased concentration of calcium in the cytosol or the cytoplasm. Okay. Now this increased concentration of the calcium results in activation of cellular enzymes. So what are the enzymes? Phospholipase, protease, endonuclease and ATPase. So as the name suggests, these are all for lysis. So how does phospholipase act? It reduces phospholipids. Protease will cause disruption of the membrane and cytoskeletal proteins, resulting in membrane damage. The endonuclease will cause nuclear damage and ATPase will cause further depletion of ATP. So this reduced ATP will increase the mitochondrial permeability transition. So this mitochondrial permeability transition will again cause or facilitate movement of calciums from the mitochondria into the cytosol, and this vicious cycle is going to repeat itself. Okay, so these are the changes of irreversible cell injury. So the two slides, like the previous one and this one, they have covered 2 points. One is the changes, pathogenesis and changes which happens in irreversible and reversible cell injury, and also the role of calcium in cell injury. Could you appreciate it? Yeah, these 2 points are important. Okay. Majority of the injurious agents, they affect the cell by the previously mentioned 2 methods. But free radicals, they have what they cause cell injury in a very distinct fashion. Okay, we'll learn it. Now first we'll know what is a free radical. Anybody knows what is a free radical? Reactive oxygen species is a very good example. So what is their characteristic? So these are chemical species which have a single unpaired electron in their outer orbit. So if they have a single unpaired electron, that means they are deficient in energy. So how do they act? They are like robbers. So what they do is, they go and attack the normal cells and they try to snatch away one more electron for pairing, and so that they can become inactive. So what are the examples? Superoxide oxygen, hydroxyl radical, hydrogen peroxide, these are the common ones, and nitric oxide, peroxynitride and hypochlorous acid. So all these are examples of free radicals. So how are free radicals formed? So they can be formed from a lot of sources. One is UV light, then ionizing radiation, smoking, exposure to air pollution, from inflammation, and also from the mitochondria. So injury to the mitochondria can give rise to what? What are we studying? Free radicals. So why is it important? Why should we know free radicals? Because they are associated with cell injury, especially caused by chemicals, reperfusion injury, inflammation, irradiation, oxygen toxicity and also carcinogenesis. So how do these free radicals cause cell injury? So taking hydroxyl free radical as the example, because it is the most reactive and most potent, they cause lipid peroxidation, protein oxidation, DNA damage and cytoskeletal damage. All of these changes will lead to cell death. Okay, so free radicals usually cause what kind of cell injury? Cell death is the result of what kind of cell injury? Irreversible cell injury. So we have seen from the previous slide, right, that free radicals are formed from many sources. So with that fact, it should happen that our body will be having a lot of free radicals and it should be affecting all the cells. But does it affect? No. So how is it warded off? Our body has its own mechanism to prevent or to neutralize the free radicals. What are they? Antioxidants. So these can be endogenous or exogenous substances which will inactivate the free radicals. So how do they act? These antioxidants, they will have an extra electron which they donate to the free radicals, and the unpaired electron in the free radical becomes paired now, and the free radicals become stable. So the few examples are vitamins A, C, E, cysteine, glutathione peroxidase, cellular plasmin and transferrin. So these examples you have to remember. Okay. Now with the concept of cell injury and the concept of free radical injury, we have to understand ischemia-reperfusion injury. So this ischemia-reperfusion injury is basically the good, bad and the ugly. Can anybody tell me which is the good part in it? Okay, we'll first start with the bad. Which is the bad part in it? What is the bad part in ischemia-reperfusion injury? Ugly is the worst kind of bad, right? So which is the lesser bad here? What do you think? It's not a test, just what do you think? Yeah, you're correct. So bad is the ischemia, which is the good. Reperfusion is the good, and obviously the ugly is the injury associated with it. So the bad is the ischemia. So what happens in ischemia? There is occlusion of the coronary artery, leading to reduced blood supply to the cardiac muscles, and which can result in death of the cardiac muscles. So the good is the reperfusion, which is the reopening of the coronary artery, which can be done by a lot of ways, one of which is using a balloon angioplasty. And the ugly is the reperfusion injury. So we do it like the reperfusion is done with a good intention, but sometimes it can result in the ugly outcome of reperfusion injury, which is irreversible cell injury leading to cell death. So how does it happen? So here we know from ischemia there will be reduced ATP and increase in the sodium and calcium concentration within the cytoplasm. So up until here it is the steps of what kind of injury? Reversible cell injury. So the reperfusion has to be done when the cells are in the reversible cell injury phase. Otherwise, it will result in worse. So once the reperfusion is reinstated, what happens? If few of the cells have already moved on into the phase of irreversible cell injury, they cause generation of free radicals. Okay? So these free radicals will further cause mitochondrial damage, leading to increased movement of calcium into the cytoplasm and resulting in cell death. Okay? Do you guys understand this? But the cells which are in the phase of reversible cell injury, they will have mitochondrial membrane recovery, leading to survival of the cell. Okay? Any confusion regarding this? Yeah? I feel like all of you are confused. Should I repeat it? Yeah? Okay. So here we are saying that up until the increased concentration of sodium and calcium, these are the stages of reversible cell injury. So from here the cells can go back to their normal functioning. So when the reperfusion is done at this phase, in case if most of the cells are in their reversible cell injury form, the mitochondrial damage is going to be repaired. Okay? And the cells will undergo survival. But there will be few cells, or there are cells which will be in their state of irreversible cell injury. Did you understand this point? Like, it is not homogeneous. Okay, if we've had an attack for 21 minutes or 22 minutes or something of that sort, so we can expect few of the cells to have moved on to their stage of irreversible cell injury because of the presence of stressor agent. No? Are you guys understanding? Let me know, give me some cues, otherwise I'll repeat it. Is it clear till now? So in those cases, what will happen? There will be generation of free radical from the affected cells. Okay? So this free radical generation, what will it do? It will affect the mitochondrial membrane. So there will be damage. So what happens if the mitochondrial membrane is damaged? What moves out of mitochondria? Into the cytosol. What is the most important ion which mediates the injury? Calcium. So there will be further increase in cytosolic concentration of calcium, leading to mitochondrial damage and cell death. So if there is cell death, there will be infarction. Okay. Now is it okay? Better? Okay. So we have to remember two factors which will determine the outcome of ischemia and reperfusion injury. One is the duration of ischemia. So if it is a longer duration, if the patient have had a longer duration of ischemia, then the chances of ischemia-reperfusion injury will increase. And the collateral circulation formation. So if there is an establishment of a collateral circulation, then what will happen? Will there be more injury or less injury? More injury or less injury? Collateral circulation means? Yeah, making an alternate route. So if the body itself finds an alternate route to provide blood supply to the affected area, what will happen? There will be lesser injury because we are removing the stressing agent, which is the ischemia. Understood? Okay. So this is just an overview. You don't have to remember everything. Just remember that there will be oxidative stress and pH correction, both of which will lead to calcium overload. So what will happen? This calcium overload will also cause hypercontracture and mitochondrial transport channel opening, leading to lethal myocardial injury. This is more important. You remember this one. So now we'll move on to morphology of cell injury. So this is, first we'll know what are the changes which happens in reversible cell injury. So in a normal cell, we are seeing here that in reversible cell injury, it can also recover and go back to its normal homeostasis. So what are the changes? The myelin figures will be affected. Then there will be swelling of the endoplasmic reticulum and mitochondria, and also formation of membrane blebs. All of this happens because of what? What was the first step? Dysfunctionality of the sodium pump and increased concentration of sodium, water and calcium. All of this will lead to swelling and hydropic change. So up until here is changes of reversible cell injury. Okay. Now if there is persistence of the stressor agent and there is progressive injury, there are certain changes which happens in the irreversible cell injury, mainly resulting in necrosis. So what will happen? There will be inflammation, then breakdown of the plasma membranes, organelles and nucleus, and leakage of the contents within the cytoplasm, and amorphous densities will be formed within the mitochondria. So this part, or the changes which happens in necrosis and apoptosis, all of this will be dealt with in the subsequent classes. Okay. So now when we compare these changes with what happens in apoptosis, what is apoptosis? Programmed cell death. So it is a normal functionality of our body, right? So what happens in apoptosis? There will be condensation of the chromatids and there will be formation of membrane blebs as well. So these membrane blebs will result in cellular fragmentations, and these fragments are known as apoptotic bodies, which will be taken up by the phagocytes. So you could appreciate the difference between the changes which happens in cell injury and apoptosis. Yeah? Okay. So we have seen what are the changes that happens at the cellular level. So how do we appreciate it? We have to see those changes in microscope as well, right? Otherwise we will not know. So how do these changes represent itself? Hydropic change, hyaline change, mucoid change, and intracellular accumulations. So what is hydropic change? So there is accumulation of water in the cytoplasm, usually seen as cloudy swelling or vacuolar degeneration, and it is almost always the first manifestation in all forms of cell injury, whether it's reversible or irreversible. And it happens because of impaired regulation of sodium and potassium on cell membrane. We know that, we have read it. So how does it look? So this is a schematic diagram and a microphotograph of which organ do you think? What is this? This is a glomeruli and these are renal tubules. So kidney. So what is the renal tubular cells? What kind of cytoplasm do they usually have? They'll have a eosinophilic cytoplasm, but in cases of hydropic change, can you appreciate the clearing here? The entire cytoplasm being replaced by clear vacuoles? Yes? No? Can you appreciate it? Is it visible? Like see, can you see this? The cell membranes, they have also become indistinct and the cytoplasm is entirely replaced by vacuoles. Okay, here you can appreciate it a little better. So here can you see the normal hepatocytes? Yeah? Yes? Yes, right. So when you compare the normal hepatocytes to this area of the hepatocytes, what do you see? What is the difference? First is that they have lost their uniformity. They are not of same size. Few of the cells have become very large. And their cytoplasm, it's no more eosinophilic, it's replaced by vacuoles. Can you appreciate this change? So this is known as ballooning degeneration. This is also associated with toxic injury to the liver. Next is the hyaline change or hyaline degeneration. So hyaline basically means glassy and it appears as homogeneous eosinophilic appearance of proteinaceous material, and we can appreciate it in the H&E stain itself, and it does not refer to any specific substance. It can be intracellular or extracellular. So intracellular hyaline, it is majorly seen in epithelial cells. So this is an example of hyaline droplets. So now can you tell me which organ is this? We've seen this just two slides before. This is a glomeruli. These are renal tubules. Yeah? Can you appreciate that this is a kidney? Okay. So don't confuse these. These are RBCs. Can you see these eosinophilic droplets? Can you see? Can you appreciate them? Yeah? So these are hyaline droplets. So why are they formed? They are formed in cases of proteinuria, when there is increased reabsorption of protein by the renal tubular cells. Now this is an example of hyaline degeneration of the muscle, especially the rectus abdominis muscle. This is a very specific name called Zenker's degeneration, and we see it in typhoid fever. And this is in case of alcoholic liver cell injury. Can you see it in the inset here? Collection of eosinophilic material. All of you, can you see it? These are called Mallory bodies. Now we also have Russell bodies as an example. So this is a plasma cell. Okay, and when there is accumulation of increased immunoglobulin within the cytoplasm, there is formation of Russell bodies. Yeah. Okay. So extracellular hyaline is seen when is present in the connective tissue. One example is hyaline degeneration of leiomyomas. What is a leiomyoma? Are you aware of the more common term fibroid? Yeah? No? No? So it is a benign neoplasm of uterus. So here you can see that these are a molding pattern of the cells is a very characteristic feature of leiomyoma. So the surrounding here shows molding pattern, and here you can see a glassy eosinophilic material deposition. This is example of hyaline degeneration. Then we also have examples of hyalinized old scars, and a very important example is hyaline arteriosclerosis. Here you can appreciate, right? It's pretty evident. Yeah? So this changes is seen in cases of hypertension and diabetes mellitus. Now another change is mucoid or myxoid change. What does mucoid or myxoid mean? Are the same. Mucoid itself means mucin-like. Even myxoid means mucin-like. So what is the difference between mucoid and myxoid? Any idea? So basically it's this difference in the origin. Mucoid is when there is production of mucin by the epithelial cells, and myxoid is when it is in the connective tissue. So this here depicts, see they almost look similar, right? So morphologically we will not appreciate much of a difference, but it is just the tissue in which it is present. So when it is present in the epithelial region, like in the intima and the media in peripheral artery, we call it mucoid change. But here you can see when it is in the stroma, so it is the stromal part of a fibroadenoma. When it shows mucin-like changes, we call it myxoid change. Now what are the morphologic forms of irreversible cell injury? Autolysis, necrosis and apoptosis. So these three will be dealt in subsequent classes, and also intracellular accumulations. Intracellular accumulations is a feature of irreversible cell injury, but we'll deal it in details in the next class. So from what we have learned today, you should be able to answer these questions, okay? Definition of cell injury, different types of cell injury with examples of reversible and irreversible cell injury. Then the explain with diagram the response of cell in response to stress and injurious stimulus. You remember the diagram, the flowchart? Okay. Then role of calcium in cell injury and ischemia-Reperfusion injury. Okay. So do you have any questions? No? I thought so. We'll go to the MCQs. Okay, today I want volunteers to, stand up and explain the case-based questions to me. So first one is pretty recall-based. It doesn't matter, like, which of the following is a common cause of cell injury? It's hypoxia, correct? Okay, now second one, who will answer? We'll start from the last bench. It's always fun to start from last bench. Yes, yourself. You explain to me. So a pathologist examines a liver biopsy from a patient with chronic alcohol abuse. Light microscopy shows ballooning degeneration of hepatocytes, which are swollen cells with large cytoplasmic vacuoles and some fat accumulation in the cytoplasm. So which of the following is the most likely mechanism of this change? Hydropic change. So hydropic change is a feature of what kind of cell injury? Reversible cell injury. Was it a guess? No? Okay. So hydropic change. Third question, sit. Who will do it? Yes, this side. Extreme left. Yourself. Yes. So wait. A patient presents to the emergency room with severe substernal chest pain, which is lasting for 4 hours. Blood tests reveal a significant elevation in serum creatine kinase levels. So what is the most likely cause of the elevated serum CK? A. Because of a small increase in mitochondrial activity. It is, the answer is C only. Very happy with the guesses. Okay. So this is a case of myocardial infarction. So from MI we know there is cell injury going on. So which is a feature of cell injury here? damage to the membranes. So if only there is damage to the membranes, there will be release of enzymes. Correct? So it is going to be damage to the membrane of myocytes. Can you guys see the board? I mean, like, it's visible, right? Okay. Okay, fourth. Who will answer? Okay, this side. I'll read out so that everybody can hear. In an experiment, cells are subjected to radiant energy in the form of X-rays. This results in cell injury. Which of the following cellular enzymes protects the cells from this type of cell injury? B. Why? Yes? Yeah. So what is glutathione peroxidase? Correct. Good. So any confusion with any of the other questions? Any doubts? All right. And I thought so.